John, thank you very much, and also express my gratitude to the organizing groups to allow somebody out of state to come up here and just briefly talk. Mostly talk, I'll hand over to Dr. Highland here in a minute. But just give, many of you know about the U.S. Sheep Experiment Station in Du Bois, Idaho. We are a USDA, based in the USDA, specifically the Agricultural Research Service Agency. Those that are not quite familiar with us, I just want to give you a brief introduction of how we're set up and how we're designed that complements, as far as our co-partner in research, specifically on the disease side, would be the group out of Pullman, the ARS lab out of Pullman, with Dr. Knowles and Highland. So we are set up to mimic a rangeland-based sheep production system. Dave, one thing, Dave, you said you'd forgot. There was something when we was talking about USDA and Montana State. He was like, there was one thing I forgot. It'd be Targees and Columbias and poly-phase sheep. So those particular breeds were developed and released from the U.S. Sheep Experiment Station over the past 90 years, and which Targee is one of the popular breeds that you see in the state of Wyoming. So again, in mimicking that rangeland sheep production system, about 2/3 of the nutrition, the way that we manage our sheep, it's commercial life, but we have that research component that envelops that. So 2/3 of the nutrition for our sheep is derived from rangeland, much like you would see in the Upper West sheep production systems. Primarily, we graze open rangeland, sagebrush communities, specifically big sage and three-tip sagebrush communities. We are a shed landing system. So throughout Montana, you'll see somewhat of a variety of small pasture-based systems landing out there, where we mimic a lot of the larger type production systems where they're landing indoors, where the sheep are indoors maybe for 24 to 48 hours. So it gives us a lot of access in terms of when we're tracking and monitoring pathogens or whatever it may be in our types of experiments. The main base of our flock is the wool-type flock of what you do see here in Montana. We also have the semi-prolific breeds, the composites like the polypay, and also the terminal sire breeds like the blackface sheep, the suckle. So we do research with those because those are also an important component of the sheep industry, especially in the West, as far as crossing part of their flock to the terminal type to produce a premier carcass or something that the consumer is looking for. Oops, I guess I should hit the button. Well, I'll hit the I'm sorry, John. How's that work? That'll work. Okay. So health-related, we have a number of research objectives that go across range ecology, rangeland science, but specific for this particular event. Our health-related topics that we focus on is, and Dave mentioned, in terms of sheep production, we are very concerned about health. We're very concerned about the mortality and morbidity rates of whatever disease or organisms that may be in that specific flock. So a lot of our research on a production-based environment that we focus on the U.S. Sheep Experiment Station are looking at antibiotic alternatives. I mean, you as a consumer as well as regulatory, we can see that in the United States, much like in Europe and other parts of the country, is that whether it's by regulation or you as a consumer in terms of the products, the meat products that you consume, is that we see a lot of the industries, a lot of the animal food animal industries going more and more towards we're seeing less antibiotic usage in those particular production systems. So we have to look for those alternatives, whether it be management or something that's a non-antibiotic type compound. Also vaccination strategies. And we have a number of vaccinations against a number of different organisms and viruses that do infect our sheep. So it's working with the producer and looking at different strategies that result in the best type of vaccination or get the best efficacy out of that particular program that they're applying to their health. And then neonatal health. I mean, we'll hear it on both sides, no matter what it is, whether we're talking about mammals or avians. That young animal, that's where most of the death will occur, no matter whether you're talking about a domestic or a wild species, that neonatal period. So a lot of our focus is there. Also, reproduction longevity, as far as having that flock out there, having that flock produce the most efficient that it can for the resource base that it's using out there on the rangelands, is getting the most over a lifetime from that particular ewe, and that relates a lot to health. And also flock health monitoring. And that's where, I'll turn it here in a minute over to Dr. Highland, is at the U.S. Sheep Experiment Station. Our basis is production, genomics, genetics type research geared towards that. And then our co-laboratory, or our co-research, I guess you could say, at Animal Disease Research Unit at Plymouth, Dr. Knowles and Highland, handle the specifics on the disease, whether it's monitoring, surveilling, or looking at maybe looking across the genome to try to find, are there any traits that may be associated with some degree of disease resistance. So, Maggie, she's going to carry the rest of it.
Okay. This moves it then? Okay. So I'll start with this statement. It's talking about pneumonia in general in small domestic ruminants, be it wild or domestic, although this statement is based on what we think we know about domestic sheep pneumonia. So this basically just talks about how complex of an issue pneumonia is, even in domestic sheep, and I think that reflects really well in wild sheep, and that's why for so long it's taken us so much work and research and further research to understand the disease complex in bighorn sheep. So just an overview, what I consider the foundation of infectious disease. To start with, I like to now talk about, whenever I give presentations, that diseases are not transmitted. That's a misnomer. Whoever started saying that was way off base. Diseases are not transmitted. Infectious agents are transmitted. Disease is the outcome of transmission and is dependent on really 3 factors. What I had learned in veterinary medical school to be the beast, the bug, and the burden, and where those 3 overlap is when disease happens. So we've looked extensively at what we consider, I don't know how that works, but consider to be the bug that causes pneumonia in bighorn sheep and in domestic sheep. We are looking more now at the beast itself, considering its nutrition, and maybe genetic factors that can be associated with susceptibility. And then the third thing, which I think is now being more looked at, as I believe John just talked about, is the burden, which is like the environment, which are the stressors that are put upon an animal that allow these all 3 to come together and cause disease in an animal. So I was asked just to give an overview of the current and upcoming research that we do at ADRU. Our national project plan, this is our next 5-year budget for doing research, and our stakeholders guide us, and national programs guide us on what our research is going to be. And so the title of our new research project is Identification of Host Factors in Immunopathogenesis of Pneumonia in Domestic and Bighorn Sheep. And there's a lot of words on this slide, but basically our project is going to focus most prominently on Mycoplasma ovipneumoniae. This bug has been discovered in the last decade to be highly associated with the complex phenomenon of bighorn sheep pneumonia, and large credit goes to Dr. Tom Besser for discovering this organism and really advancing the research and identifying this bug as a primary agent. Tom's with us today. So this organism impacts adults and lambs, but not consistently. So infection doesn't equal disease in bighorns or in domestic small ruminants. The pastorellas and other mixed bacteria that are often found in association with pneumonia in bighorn sheep and in domestic sheep. And for a long time, it was the pastorellas that were really thought to be the primary agents, but they're not, the individuals, genus and species of pastorella are not consistently identified. certainly not as consistently as Mycoplasma pneumoniae is in bighorn sheep pneumonia. So it just kind of substantiates probably the primary role and makes Mycoplasma pneumoniae the most sound epidemiological organism associated with bighorn sheep pneumonia. It's been known for decades, this organism has been known for decades to be associated with domestic sheep and goat pneumonia. It's often associated with suboptimal environmental conditions, as Dave McEwen in his talk already talked about. In domestic small ruminants, it primarily affects lambs and kids. Infection can result in lowered lamb production, which has been reported in a brief communication from New Zealand. And it's present in many domestic sheep flocks across the United States, as shown by a National Animal Health Monitoring Service study that was performed. And then it's also present in some domestic goat tribes or herds, making this an endemic bacteria in the US. It's believed to be species specific currently, affecting or infecting only members of the Caprinae subfamily, which are goats and sheep. So just a brief overview of the project plan, the research that we're going to be diving into and are already in the progress of doing. So we're looking at host genetics of infection and shedding of Mycoplasma pneumoniae in domestic and bighorn sheep. We're also looking at innate and adaptive immune factors associated with susceptibility of domestic and bighorn sheep to this organism. This will include the immunopathogenesis, meaning just how each species responds to exposure or infection with Mycoplasma pneumoniae, looking at both innate and adaptive responses. And then towards the end of our project plan, we have work that we're going to be doing in potentially developing a vaccine, whether it be for the domestic or wildlife side, but focusing more on domestic side. So additional research that we're working on is nasal or respiratory microbiota of small ruminants. Also surveillance of non-Caprinae species for carrying or shedding Mycoplasma pneumoniae. I've currently looked at quite a few elk, a few whitetail. Whenever I can get a hunter to stick a nasal swab up the nose of a whitetail deer. And that's about the limit thus far on what we've looked at. And to date have not found MOV, but like I said, we're really limited in the number of animals that we've looked at, and the elk that I tested were just from one privately owned herd. And then also surveillance of pack goats and non-packing domestic goats for Mycoplasma pneumoniae prevalence, as well as agents, carrying status for agents that cause keratoconjunctivitis or pink eye that are considered to be a risk for bighorn sheep. And that project is near completion, and that was started because there were concerns I was contacted by the North American Pack Goat Association because there were concerns that they were concerned about domestic goats posing a risk to bighorns and all the bans that were being imposed upon them for using their packing animals on public lands that are deemed bighorn sheep habitat. The concern comes from the fact that goats can carry bacteria that are associated with bighorn sheep pneumonia, and goats can carry bacteria that are known to cause pink eye. And I just described here that how NAPGA had contacted us to try to figure out if we could do a broad-scale surveillance study. So to get through this kind of quickly, for time's sake, we tested a total of 576 goats with 3 nasal swabs being collected at intervals of 4 weeks or greater time periods. 419 were considered packers or future packers. So these were young that were being raised to be packers, and another 157 that were on the premises. They're non-packers, so they're milkers or just pet goats that are on the same premises. And as I just briefly mentioned, we collected 3 sets of duplicate nasal swabs at 4-week minimum intervals, as well as a set of ocular swabs, 1 swab from each of the eye, and that was collected just once, and then we collected blood and serum from every animal. Let's see, and then we processed all samples within 72 hours of collection. Duplicate nasal swabs from the first sample collection were submitted to the Washington Animal Disease Diagnostic Laboratory for confirmation from an independent laboratory outside of ADRU. And then our swab samples, both nasal and ocular, the nasals we finished testing those and we're still looking at the ocular, but we're tested by PCR and or qPCR, and then the positive samples were confirmed by DNA sequencing from PCR products. Let's see. So here are the results. This is the Waddle results, so the diagnostic lab results. They had, so this is the number of animals and the number of premises. They showed out of the total tested, so they tested 468 goats. Waddle did not test all of the non-packers on the premises. They identified MOV in 18 of the 468 goats tested, which came from 5 different premises. And then indeterminate results from an additional 20 animals on 9 premises, of which these overlap. So just 4 additional. And then not detected, where they didn't detect any MOV on the nasal swab in 430 of the 468 goat sampled. So that leads to, at the highest would be these 2 added together, considering all the indeterminates, if they were actually positives, at just under 10% positives for the overall animals that were tested. So a fairly low prevalence. To go beyond all these words for right now, this is testing that was done by standard PCR in our laboratory, and then we did follow up by sequencing to confirm whether the product, the PCR product, was really Mycoplasma pneumoniae. We confirmed that out of 3 of the 5 premises that there was the presence of Mycoplasma pneumoniae in these goats at 2.8% of the animals tested overall, and then the not detected over here. So we are continuing to work to do additional qPCR and PCR on nasal swab samples to focus on those that were detected that we were unable to confirm in our laboratory, but Waddle had detected, or even the indeterminates. We're trying our best to make sure that when we say that the animals are negative, they are really negative. And then we also have to work on PCR, qPCR, the ocular swabs yet, so none of that is done. But just overall, a very low prevalence, and that is in vast contrast to a recent publication out of Washington State regarding prevalence of sheep and or goats having 30+% prevalence of MOV. So this is vastly different with a lot more animals being tested from throughout the West. And of interest, the animals that were confirmed positive on 2 of the 3 premises were under 12 weeks of age. So I'm interested in looking a little bit more on that, for at least for kid goats. I know this is more of a wool growers, but sheep and goats are grouped together often now. So, and I don't know if that's appropriate as far as looking at Mycoplasma pneumoniae. So this might bring up controversy, but I don't know how to talk about this topic without bringing up controversy. And I think folks today have done a really good job in their speak, but often the media, and I know a lot of us cannot control what the media prints, even if it's not what we actually say. I've had it happen to me. And it doesn't matter if you yell at them, they'll do it again. But when we have publications that are peer-reviewed and you find biased or strong sentiment that isn't exactly true, it does no justice to research. So I'm going to give an example here. So this was taken from the introduction of a recent publication. So it says, there is now clear evidence that polymicrobial pneumonia is often facilitated by spillover of Mycoplasma pneumoniae. And the paper was about spillover from domestic sheep and goats. It says, Mycoplasma pneumoniae, a common bacterium of both domestic sheep and goats that rarely produces clinical disease in domestic caprinae, that being sheep and goats. And as Dave just alluded to, given the right environmental, or actually the incorrect environment to put domestic animals into, it does cause quite severe disease in domestic animals also. And if you look up the references that are used here, this was from Nicholas et al. 2008. And this is out of their introduction that's referenced here. And it says, compared to other ruminants like cattle and goats, sheep are not as prone to the same range or severity of mycoplasmal infections. So again, they even separate out sheep and goats, so making this statement not quite true. And then in this same paper, Nicholas et al. Goes on to say, however, the role of mycoplasmas, in particular Mycoplasma pneumoniae, is often overlooked, and as well as causing disease in its own right, it can predispose animals to pasteurellosis and viral infections. So again, they're talking about domestic sheep here, just to again say this is a problem in the domestic sheep industry also, and it really parallels, I think, when I read about bighorn sheep pneumonia and talk to producers, and when I read about domestic sheep pneumonia, there's quite a few parallels actually in the disease complex of pneumonia in small ruminants, be it wild or domestic. And the other two publications, these are just prevalence studies that were done on domestic sheep only. So this whole sentence would allude to would make the reader believe that the clear evidence is found in these references, which it's not, and it also would indicate that mycoplasma pneumonia is not a problem for small domestic ruminants, which it is. So it's just those kind of statements. I would just ask everybody that works on this problem to be as unbiased in what you say, because when you make statements like this, it almost makes the other side, which there shouldn't be another side, we should work together, but it makes the other side think that you're less credible, or that you're making stuff up, or that you're trying to push an agenda, and it leads to distrust. I think it's led to a lot of distrust in this problem. And again, I'll come back to the foundation of infectious disease and what we really need to focus on. And there's a lot of people to thank. A couple are in the audience here, Bret Taylor, Whit Stewart, who we collaborate with. All of the work that was done for the pack goats was headed up by Diane Norton, and a really long list of wonderful APHIS people that collected these swabs in a very short amount of time from the goats, and then my contact info. That's it.
Referred to them always, and I understand that now it is in. Oh, excuse me. I was violating my own rules there, wasn't I? Okay, thank you. Thank you, Dr. Highland. And you alluded, or you said that, and I understand that now MOV is endemic in the United States. Do we know, and maybe nobody does, but was it here natively, historically, before we brought over all the other, livestock that came over with settlers and et cetera?
I think there's indication that it was not, and I wonder if Dr. Besser's not going to speak to that a little bit. Yeah. You're not at all? No? Well,
Nobody would know.
It's hard to prove that. Yeah. Certainly it is. I mean, what Europeans brought diseases to Homo sapiens brought diseases with them too, right? So things that others have never been exposed to. And I think one of the big things to focus on is, okay, we know some of the sources. The ones that we know are sources of mycoplasma pneumonia. It's why there hasn't been some development of like a genetic move in bighorn sheep to handle it. But we know it's not a killer for all of them. So why do the survivors survive? And I think a lot of that might go into the environmental or the burden component that's being looked at.
Yeah, and in some ways it's an academic question really, and we know that it's here now and we have to manage for it. So.
Yeah, and endemic is quite, people would question that word too probably, but as far as or geographically, it is present in the United States in large numbers of animals.
I have a question real quick. Thank you for the presentation. I'm trying to wrap my head around a couple of the concepts you mentioned, and so hopefully you can explain it. You said infection does not equal disease, but then you said infection on its own can lower lamb production. So I'm struggling with how that works.
So subclinical disease. So an animal can be a carrier, so they're infected, an organism is actively reproducing itself within some part of the body, let's say the nasal passage. In domestic lambs, you can see a subclinical pneumonia, that is regional in a lung lobe only. So you wouldn't know that it's a disease looking at them clinically, but at slaughter or some other event that kills them, if you open them up, you'll see typically one lung lobe, and it's their accessory lobe. It's the catching lobe, I call it. It's where bacteria kind of fall. It's the first branch off going down the trachea. So they have a subclinical disease, potentially, that's not being recognized from Mycoplasma pneumoniae, which typically is not seen then in adults. So even those that are grown to be for wool or the ewes that are grown to be used further, I don't see that lobar type of pneumonia in them, even though they may still carry Mycoplasma pneumoniae.
So they can have that and their lambs can die even though they are technically healthy, or I mean they're infected but not.
So the ewe can have it, the lamb, I think, again, Dr. Besser's doing work on some of the transmission in lambs and at what time they become infected. I don't know if that lobar pneumonia occurs at the waning of passive transfer or at what point. I don't know that there's been a time necessarily, but I don't know that whether or not lambs necessarily become infected at an age as early as bighorn lambs do is one of the comments that I've seen Dr. Besser mention in his presentations too. And I think more work needs to be done to look into that, but there is subclinical disease also. So infection in those lambs, when it causes a lobar pneumonia, has been associated, a New Zealand report that had come out, with decreased production in lambs that have that clinical finding, or the autopsy finding at the time of slaughter.
I just have a quick statement. I just want to second what Maggie said about us being more respectful to each other. Unfortunately, in many instances, we still do have major sides, and I want to point out that it is up to all of us to make sure that either side does not put out false statements, and that the only way we are going to try to get together is to try to find common ground and not berate each other in public or in private, and just be a little bit more respectful and try to find the commonality in this issue and not always look for the differences. So, thank you.
Maggie, in the spirit of the meeting, how do you proceed moving? This is Whit.
I'm like, I know you're here. Where are you?
Sorry. Is there an opportunity for greater collaborations to exist or greater information sharing? Do you feel that the opportunity to share more transparent information, all these other factors that are related to the presence and manifestation of disease, how do we overcome that barrier? Because we all live it within state lines and we have our camps. How do we get over that?
Something I used to have at the end of a lot of my slides was to try to figure out a way to put a database together where everybody comes together and having conversations. I hear that somebody has actually started that in the last year or so. But I don't know conversations with Dr. Ramsey in Owensburg. But I don't know who's going to have access to that, or, you know, if you call and ask a question, have there been any introductions? Have you done recent captures in that area? Things like that, I think, should be anybody's knowledge. That's just my personal opinion. I don't know why there would be any hidden information about health of bighorn sheep or where they're located, or what their infection status is for Mycoplasma pneumoniae, or any of that. So, but I do agree that I think a publicly shared database, at least for all entities that are involved in the research component, would be a wonderful thing. And it needs to be from both sides that do still exist. So it's not just, you know, one arm of this problem being looked at.
Thank you.
Okay.